Oh, hi. I didn't know I was on mute. Good afternoon. Thank you for being on time. Our lesson for today is going to be on biotechnology. As I was saying last week, this is a particularly difficult topic. That's why we are having a synchronous class to just make sure that you guys are able to, learn it um, firsthand. And if you have any questions, you are able to ask me immediately. So we will meet again on Wednesday to finish up, but let's see how much we can done for today's class. So what is biotechnology? Biotechnology is, exactly what the word sounds like. It's technology that's based on biology, right? A lot of times we understand various cellular processes that are going on. For example, DNA replication. We understand how your cells do DNA damage, and we try to harness those cellular processes and biomolecular processes to develop some kind of technologies that would help improve our lives. So let's take a look at recombinant DNA as the first topic. Recombinant DNA basically means taking DNA strands from two different sources. Okay, often it's two different organisms, and then you put it together into one continuous sequence. So it's like, copy and paste. You take a sequence from a jellyfish, and then you take a sequence from a mouse, and then you put it together in one. That's called a recombinant DNA. And being able to create recombinant DNA is at the core of a lot of the biotechnology that we will be studying in this particular unit. And we can use these to in various applications, including medicine, as well as agriculture. I'm going to give you some examples of those as we progress through the slide. So here you can see a genetically modified mice here that are glowing green in the dark. Okay, so what they did is they took a green fluorescent protein, GFP, from jellyfish, and then they incorporated it into mouse DNA, and that allows them to have this greenish glow in the dark ability. And you might wonder what is the point of doing that? Like, does it actually serve any purpose? It does. GFP recombination, the green fluorescent protein, is used in a lot of research to study expression of genes, for example. We're not going to have a lot of time to go into the details, but that's just like a brief example of applications of recombinant DNA. So let's start with an example in agriculture, and then we're going to go into more details about how to apply it in medicine. Okay, there is something called golden rice. I'm not sure if anyone has heard of that before, but vitamin A is an important nutrient, if you would, for various reasons, including healthy eyesight. Okay, so there is a molecule that you make in the eye that allows you to see in low light setting. Okay, so when it's really dark, you have these rod cells in your eyes that are used for seeing things at night. Okay, and the molecules that they use is going to require vitamin A as a like a building block to make. So if a person does not get enough vitamin A, okay, and this is a common thing, deficiency in vitamin A is a common thing in countries that are poor, that could cause nighttime blindness. Okay, so you okay to see it during the day when it's bright, but once it gets dark, because you're not able to make that molecule that allows you to see in the dark, you start to become blind at night. That's a problem. And prolonged vitamin A deficiency can actually lead to complete blindness. Okay, so not only, like, you gotta learn this like semesters, but the The rod cells that is required to see at night are also needed for peripheral vision. So, if you lack the vitamin A, um, to make the proteins that's necessary for those cells to function, not only will you have nighttime blindness, you're also going to have blindness in general. So vitamin A deficiency is the leading cause of preventable childhood blindness all over the world. Okay, but particularly a problem for poor countries that doesn't have access to vitamin A. Okay, so there are other problems in poor countries, right? Including like low vaccination rate and infectious disease, poor infrastructure, and all these things just kind of amplify the effect of these blindness situation. So in some countries, each year is estimated to kill 670,000 children under the age of 5, and then we have an additional 500,000 cases of irreversible childhood blindness, which is quite substantial. Here is a heat map that shows you where the vitamin A deficiency blindness is most severely hit, and you can see these are mostly underdeveloped countries or developing countries. They don't have the resources, or they don't have the access to nutritious food in some cases, right? It's not really a problem in North America or in well-developed countries. It's more of a so to speak, like third world problem, right? So how do we fix this problem? Well, golden rice is a genetically engineered rice that is created to make something called beta carotene. Yeah, you might have heard of beta carotene. Anybody know what kind of food we naturally find beta carotene in?
Carrots.
Yeah, carrots, exactly right. The carrots, they have a lot of beta carotene, right? But carrots is bulky, right? You can't transport carrots easily. You cannot eat a lot of it, right? Because it's nutritious, but it doesn't have all the food groups that all the building blocks that you need, right, to sustain the body. And so what they do is they make the rice, which is staple food in a lot of parts of the world, to express the beta carotene. And so, when they eat their rice, not only are they getting the carbohydrates from the rice, but they will also be getting the building block that's necessary to make vitamin A. And the way they do it is they actually take one gene called Psi from daffodils, and then they take another gene called CRTL from a type of soil bacteria. And then they incorporate it into the rice seed, which allows them to make the beta carotene. So this is an example of a recombinant DNA where you have genes, genetic material from two different sources, and then you're putting it into the rice. So there are some controversies surrounding these things. A lot of these biotechnology, right, when they first introduce this, they usually face some kind of controversy, some kind of backlash, pushback from the from society. And one of the earlier issue was that the golden rice did not actually have sufficient vitamin A. Okay, so they are expressing the beta carotene, the building blocks, but it's not at a level that's enough to remove the deficiency. And so they had to come up with like a newer version, golden rice 2.0, if you would, and that contains way more beta carotene. So that was fine. But some organizations like Greenpeace, they oppose the use of these genetically modified organisms, as they are called. These are usually patented technology. They feel like there is too much control of from these corporations, right? They're developing these GMOs as to what can be grown and what cannot be grown in certain parts of the world, and they feel that was not good, and they also feel like this opens a door to more widespread use of GMOs. Now, whether you think GMOs are good or bad, like healthy or not, that's your own opinion, but this is an example of how a GMO is able to help solve a problem, right? A problem that is caused by lack of vitamin A, and now you introduce it in a food source that is commonly being eaten to express the vitamin A. and that helps solve the problem. So other controversies, right? We're about corporate control. I talked about that already. And other argues that it is sufficient to get vitamin A from other food source, right? You don't have to, genetically express it in some non-native species. Why don't they just eat it from, the natural sources? But again, the problem is those foods are not typically available, right? It's hard to transport them to those parts of the world. And even if they're available, not everybody is going to be able to afford them. Okay, so that's the agricultural use. Now we're going to move on to the medicinal use of recombinant DNA. Okay, so this is the case of insulin. Long story short, diabetes. There are two forms of diabetes: diabetes type 1 and type 2. Okay, type 2 is the one that's related to obesity. Okay, when your lifestyle, for example, poor choice of diet, lack of exercise, for example, those things could cause you to develop diabetes later on in life. That kind of diabetes, type 2, does not require insulin shot. Diabetes, the type 1 diabetes, that's the one where you are either born with it or you develop it early on in childhood. And the body does not make insulin in that case. Okay, so for those of you who do not know, insulin is important in regulating blood sugar. When you have a meal, right, your pancreas, which is located on the upper left quadrant of your abdomen, it releases the insulin to help absorb the sugar from your blood. Okay, so your cells can use it for cellular respiration. Okay, so without the insulin, even though you're eating, but your cells are not getting the sugar. So, on a cellular level, you are starving. Right? So you need to inject insulin into these individuals. Okay. So insulin was first discovered by Frederick Banting and Charles Best. These are Canadians, and there is actually a building dedicated to them at the University of Toronto. And at the beginning, there were no good way of synthesizing, of creating insulin, okay? At the time, the primary commercial source of insulins were from cows and pigs. Right? They have to extract the insulin from these animals, and then they would, try to purify it and give it to patients who need it. So what are some drawbacks of this method? Right? If you're taking it from the cows and from pigs, what are some problems associated with this? Anybody want to give me some ideas? No problem. It's okay to just take the insulin from the cows and the pigs and give it to humans. That doesn't sound wrong to you? Okay, well, I'll tell you. Number one, there is problem with compatibility, right? Okay, the cow version, the pig version is not completely the same as the human version. Okay, so it might not work as well. Okay, it might have rejection issue. You might have shorter half-life, so you might need to inject more frequently, right? So all these things are a problem. Extraction is also a problem, right? You gotta physically take it from the cows and the pigs, and you have to purify it. It's a very lengthy process, involves a lot of money, right? So it's not cheap. You cannot mass produce them, right? Think about how many cows you need to get, right? To extract the insulin from the pancreas, right? So it's not easy. It's not simple. Right? So that method was eventually replaced by genetically engineered bacteria in the 20th century. Okay, so we will talk about how that happens. So take insulin for example, right? If we want to make insulin, which is a hormone, a protein hormone, and you should know by now at this point of the course that a protein hormone is going to be encoded within the DNA. Right? So number one, first thing we have to do is we need to locate the gene of interest. We're gonna find out in our DNA, where is this gene. Once we found the gene, we are gonna have to clone it. Okay, we're gonna have to make many copies of it, and then we're gonna insert it into a vector. I'll explain what that is later. And the vector will be fed to bacteria, and then you grow the bacteria on the dish, and then the bacteria will now produce insulin for you. Okay, so we have to find a gene, we have to copy the genes, we put it in a vector, something that we can feed to a bacteria. So now the vector contains the insulin gene, we give this to the bacteria will now become insulin factories, basically. Okay, and nature will do the rest. Someone asked me, quiz 4 is not today. No, I think quiz 4 is next week, right? Quiz 4 is next week. If you, I'll double check the online and see if the test day was set incorrectly, but it's gonna be next week on lecture 8 and 9. Okay, so let's go through these steps. First, we're gonna talk about locating the gene. The Human Genome Project was an international collaborative research program with the goal to complete mapping and understanding of all genes of human beings, right? This started long time ago, okay, in 1990s. I remember when I was in high school, my biology project, like we had to do a research project and present to the class. I did it on the Human Genome Project, and at the time they were just like starting, you know, not too long ago, right? So their goal is to map out all the genes in your genome. Okay, they want to look at all the A, C, T's, and G's that makes up each of the chromosome. They want to know where they are. They want to know what they represent, and then they want to know the variations, okay, that exist within the population. And it was eventually completed in 2003. Okay, so that took like 13 years to complete, and it's like a big international collaborative effort. So now that the HGP, Human Genome Project, is completed, we basically have the genetic blueprint of humans, and this information is freely available online if you look up genome browser. So I'm just gonna show you what that looks like here. Okay, so this is the genome browser. Okay, I can go back to the home page, and you can search up any gene that you want. Okay, we learn a few genes throughout the course. Does anybody remember the name of any genes at all that we learn in this course? Like a tumor suppressor gene. Does anybody remember the name of a tumor suppressor gene that we have learned? No? Okay, that's fine, that's fine. So that kinda, okay, yes, sure, no problem. So there's a gene called BRCA1, okay, which is for if people have a mutation in the BRCA1 gene, they are more likely to get like breast cancer. Okay, so if I search up BRCA1 gene, then I will see that it is on chromosome 17, okay, and is located on the Q arm, right? This is the centromere, right? This is the P arm, this is the Q arm, and it's going to be right here at this particular locus. Right? So I can click on the BRCA1 gene, and there are different versions of it, and I can choose the genomic sequence. Okay, and then you can change the settings. We're not going to worry about it. I'm going to click submit, and there it is. That's the gene. Right? It goes like GCT, GAG, blah 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 blah. It's just pages and pages and pages of it. This whole thing, right, codes for the instruction to make that particular protein. Right, and you would know how to do this. Like you could decode it, you could change it to RNA, right? So like GCT, this is coding strand, right? So then it would be like GCU, You can look up GCU on the codon chart, right? So that's the whole gene. Now, some of it is capital letters, some of it is small letter. Anybody want to guess why some are capital, why some are small case? What do you think?
Is it for dominance and recession?
Interesting. Good guess. But dominant and recessive is talking about the whole gene, right? It wouldn't be the individual nucleotides. But that's a good try. And why do you think a whole section of DNA has capital letter and a whole section of DNA has small letters? Anyone else want to guess? Okay. So these are the capital letters are actually the exons, and the small letters are the introns, right? These are the non-coding parts that's going to get removed. Okay. So after you go like AAG, right, you will skip all the way to over here, right? Like TTC. Right? So you see, all this information is available online. So if you want to clone a gene, if you're doing research and you want to clone a gene, all you have to do is go to the website, find out where the gene is, and then you can move on to the next step, to the cloning step. Okay, so that's a genome browser. Now comes the actual cloning. Okay, how are you going to clone that thing? So we use a technique called PCR, which is polymerase chain reaction. You probably have heard of PCR by now, right? Like when you do a COVID test, you can do an antigen test or PCR test. The PCR test is more accurate. So PCR is actually a technique that is used to clone DNA. When I say clone DNA, I mean to copy it many times. So it allows specific DNA segment to be copied many times outside of a cell in a short period of time. Okay, it literally takes 1.5 hours in some cases to make millions of copies of whatever DNA segment you want to make. It's extremely easy to do. You load up all the ingredients and then you put it in a PCR machine. Okay, you key in the parameters and off you go. So each cycle of PCR reaction doubles the number of copies of DNA. 30 cycles would make billions of copies, right? So let's say we have like 10 cycles, right? 10 cycles. How many copies are you gonna have after 10 cycles? If you start with 1 copy, and how many copies are you gonna have after 10 cycles? What do you think? Two to the exponent 10, right? That's 1024. Okay. And then if you do another cycle, it's gonna be 204. It just keeps on doubling and doubling, right? So by the time you do 30 cycles, that's 2 to the exponent 30, and you will have like billions of copies. So how PCR actually works is closely related to the process of DNA replication. Okay. In a cell, the DNA strands are separated using gyrase and helicase. You might remember that from lecture 6. But instead of using these enzymes in the PCR process, we are going to be using heat as a method of separating the DNA strands. Okay, so when you heat up the sample, it will cause the 2 strands to come apart. Right, the hydrogen bonds that are holding to 2 strands will be broken when you heat up the sample. And once they are separated, then we can use each strand as a template to create a complementary strand. Okay, so it's very similar to the semi-conservative model, right, that we learn in DNA replication. Now, in order to copy only what you want and not the whole thing, you would have to design a primer. Right? So this is similar to primase, right? In DNA replication, we design an RNA primer that will flank the sequence of interest. So if this is the section of a DNA that you want to amplify, you want to have a primer here and you want to have a primer here so that you will only amplify this. Okay, you only amplify this. If you put the primer a little bit further down, then you will also copy this whole section, right? The PCR mechanism will always copy everything that's between your primer, which is why you need to have your genome browser here. Okay, so one of the things you can do here is you can tell the sorry, you can tell the website that you want to include extra stuff. Like, okay, you want to include this extra 1,000 base and extra 1,000 base ahead and behind the gene. And then when you submit it, now you have all these extra things, right? So these are the flanking sequence, remember? Here's the gene, for example. Okay, and then you can, like I said, tell the website to give you extra stuff that are in front of the gene and behind the gene, and then you can use those extra stuff to design your primer, right? Okay, so once you have the primer and then you feed it to the PCR machine, then you will copy everything that's in between, right? That's why the genome browser is important, right? That's where you're gonna look up the gene, and that's where you're gonna look up how to design your primers. And once you've done that, then you know, you have to give it some polymerase. But remember, we have to heat up the DNA sample to like 90 something degrees to separate the DNA strand. And if you heat it up to that much, the protein is going to denature, right? Okay, so we cannot use regular DNA polymerase. Okay, we use something called tag polymerase. Okay, so tag polymerase is polymerase that's extracted from hot spring bacteria. Okay? Some bacteria, they live in a very, very hot environment. Okay, let me see if I can search up a picture of a hot spring. You probably have seen it on you know, TV or something. Right here. Okay, it's not coming up. Give me a second. Computer is frozen. Copy image. Huh. Fascinating. 1 second. There we go. That's a hot spring. You've seen this before, right? Like all this discoloration, right? It's because of the minerals and stuff like that. But bacteria lives in these kind of hot spring that goes as hot as like hundreds of degrees or 120 degrees, 110 degrees sometimes, right? So these bacteria, their enzymes are resistant to denaturation by heat. So we extract the polymerase from these bacteria and we use it for the PCR so that when we heat it up, they're not going to unfold. Okay. So this is the process, right? We have the template DNA. We're going to put our primers there, and then when the tag polymerase continues to build, you will clone the sequence. So there are basically 3 steps that are involved. Okay, the denaturation, that's like heating it up to allow the strands to come apart. Okay, so that's right here. Right, step 1, denaturation. The parent strands will separate because of the heat. And then you want to lower the temperature just enough so that the primers can bind to the template strand. Okay, if it's too hot, even the primers will fall apart, right? And then you want to crank up the temperature a little bit for the tag polymerase to attach to, and then clone your sequence. So after 1 round, right, you will have 2 strands. Okay. Then you repeat the process. You separate them, and each of the new strand can now be used as a template to create extra copies. So afterwards, you will have a total of 4 copies, and then 8 copies, and so on and so forth. So that's basically the concept of PCR. Okay, so I showed you how to locate a gene of interest. I showed you how to clone the DNA. The next thing we have to do is to insert the DNA into a vector. But first, let's take a look at this question and see if you can answer that. Gonna launch the poll here. Go ahead. I only have 2 people who answered. Where are the rest of you? Okay, that's fine if this is the way you guys prefer. So I have 3 different answers, 16, 40, and 80. So let's see, you have 10 copies. Okay, so if you go 1 round of PCR, you times 2, right? Okay, so you second round, third round, fourth round. So that is 10 times 2 to the 4. Right? 2 to the 4 is 16. So 16 times 10 is 160 copies. Correct answer is E. Okay. The kind of PCR test that we do for COVID testing is called RT-qPCR. Okay, Q stands for quantitative. Quantitative PCR. That means you can count the number of copies that are being created. And RT stands for reverse transcriptase. Okay, reverse transcriptase qPCR. So what does that mean? Now we know the central dogma of cell biology, right? DNA becomes RNA, which then becomes protein. Okay. The SARS-CoV-2 virus, the one that causes COVID-19, it's an RNA virus. Okay, so if you want to clone the DNA, you actually have to take the RNA from the virus and you convert it back into DNA first. Okay, so this is the opposite of transcription, which is called reverse transcription. And to do reverse transcription, you need the enzyme reverse transcriptase. Okay, so basically you collect a sample, a nasal swab. You want to if you're infected with COVID, you basically will have some viral RNA in the sample, and then you feed it into the PCR process. The reverse transcriptase will convert the viral RNA into DNA, and then you would design primers that would specifically look for the viral DNA. Okay, and if you do have the virus, then you would end up having many copies that are being copied using the RNA, using the PCR process, and that's how you know if the person has the virus or not. So this is what it looks like. A lot of times you want to track to see how many cycles it takes for the sample to reach a particular threshold. Okay, so like how long does it take for the machine to make this many copies of the viral DNA? The longer it takes to reach that threshold means the less viral particle you have. Okay, so someone who is recovering from the COVID, you would expect them to cross the threshold much later because their viral load is much lower, right? If they cross the threshold really quickly, okay, that means that they have a lot of viral RNA in their sample, right? So they are still very, very infectious. So that is useful information for doing contact tracing. Okay, like is this person someone who was infected early on and potentially spread it to many other people already, or if they're still in the early stages of infection, if we put them into isolation right away, then maybe they're not going to spread it to as many people, right? So that's RT-qPCR. RT because we have to change the viral RNA back into DNA first before you clone it, and Q, quantitative, meaning we can keep track of how many copies of DNA we are making per cycle. Okay, so we found the gene, we cloned the gene, and now we have to insert that gene into a vector, right? And the vector is often a plasmid. I told you about plasmid, some time ago. These are circular DNA that are found in the bacteria in addition to their own core DNA. Okay? And bacteria can pick up plasmids from the environment. They can pick up plasmids from dead bacteria. They could even exchange plasmid with another bacteria. So once people understood the versatility of plasmid, they were able to extract the plasmid from a bacteria, modify the plasmid, say we insert the insulin gene into the plasmid, and then we feed it back to the bacteria so that it is going to create insulin for us or whatever other protein we wanted to create. So how do we insert the gene of interest into the plasmid? Well, to understand that, we need to learn something called restriction enzymes. Okay, so restriction enzymes are actually, they originate from bacteria as well. Okay, so these are enzymes that are present in different kinds of bacteria, and the function is to act as a defense mechanism. Okay, so even though bacteria can pick up plasma from the environment, but sometimes they pick up some junk DNA that they don't want to have. So the restriction enzymes are there to cut those junk DNA and basically break them down. Okay, now the restriction enzymes don't just randomly cut the DNA at random locations. They have these very specific recognition sites that they will find and then cut. Okay, these recognition sites are usually 4 to 8 base pair long, and they are characterized by complementary palindromic sequence. Now, what does that even mean? Yeah. Does anybody know what a palindrome is in English? Just wondering.
Yeah, it means the same thing both ways.
Exactly, right? So in English, a palindrome is a word that spells the same forward and backwards. So like madam, right? Level, wow, kayak, they spell the same forward and backward. Sometimes it could even be a sentence like, was it a car or a cat I saw? If you read it backwards, it's exactly the same, right? Was it a car or a cat I saw, right? So that's palindrome. Okay, so in terms of DNA, a palindrome is a sequence that reads the same on one strand forward and the same backwards. So 5' to 3' on one end is exactly same as 5' to 3' on the other end. So GAATTC, GAATTC. So there is a palindromic sequence. And these restriction enzymes, such as ECOR-R1, that's a restriction enzyme that's taken from E. coli, they will find these palindromic sequences in a DNA segment and they will create a cut. Okay, so afterwards, the DNA separates. Okay, depends how they cut it. Sometimes they can cut zigzag like this, and it creates these sticky ends. Okay, sticky ends. That's what they're called, sticky ends, because it's possible for them to come back together. The A will be attracted to the T, and T like that, and then they will just reattach under certain conditions. Okay. So here are a bunch of restriction enzymes. ECOR-R1 is taken from E. coli. We have something like SAL1, which is taken from something called Streptomyces. It doesn't really matter where we take it from. What matters is the following. You have to know that these are restriction enzymes, and each one of them recognize a particular sequence that they will cut. Okay, so ECOR-R1 recognize the GAATTC. You don't have to memorize it. Okay, if I ask you something about our restriction enzymes on the test, I will always give you their recognition site. Here's a small one which will cut GGG-CCC into this. Okay, now there are two kinds of cuts, right? Those that create sticky ends, like that one, and that one, and these create blunt ends. Blunt ends. Right, so these are the sticky ends, sticky ends, sticky ends. In terms of inserting a gene into a plasmid, we want to use restriction enzymes that create sticky ends. the one that cuts straight down to create the blunt ends, those are not particularly useful in the context of inserting a gene into a plasmid. Okay, and I'll explain why in a second. Any question so far? Anyway, I hope this is better than having you watch the recordings. I don't know. You guys are really quiet. I can't really tell if you guys like this more. Okay, so here is a sample problem. Okay, we have a gene segment. Okay, and we are going to use small 1 as a restriction enzyme to cut this thing. Okay, so where will the cut happen? Well, all you have to do is look up GGGCCC. Now be careful, this is 5' to 3'. Okay, and here we should be looking at the 5' to 3' end for GGGCCC. Instead of looking at the top sequence, I should look at the bottom sequence. So we just go and scan it. Right, oh, and there it is, GGGCCC. So the enzyme will find this location and it will cut it right there. Okay, we're gonna keep going and we're gonna find another GGGCCC. Oh, there it is, GGGCCC, GGGCCC on the other side as well, and it will cut it like that. Right, so after using small 1, we're gonna end up having 3 separate fragments. Any question about that?
I don't understand what just happened.
You don't understand? Okay. You understand? Do you understand what this is?
Yes.
Okay. So this enzyme will look for this specific sequence. Okay. So in this whole DNA, there are 2 such locations that has the exact same sequence, right?
Oh, so it's just a tiny segment that should be palindromic,
Exactly. It
not the
doesn't have to
whole
be the
thing.
whole thing. Yeah. So then it will
Oh,
find this specific palindrome, and when it does find it, it will cut it right there. It'll cut it right there. So you will end up having 3 separate fragments. Does that make sense?
okay. Yeah,
Okay.
thank you.
Anyone else have any other questions? All right. So here, why don't you take a moment and see if you can identify the palindromic sequence? Okay. See if you can find any palindromes. If you found it, please type it in the chat box, or you can just unmute and tell me. Okay, so here is a palindrome. Okay, usually want them to be 4 to 8 segment long, right? So here, GCGC is palindrome because if you do it backwards, it's also GCGC, right? So that's a palindrome. And you can keep on scanning forward and see if you can find anything else. Okay, here's another one. There you go, that's a long one. if you write this backwards, right, it will be CTTAAG. Right? So GAATTC, GAATTC, palindromic. Anything else that you guys can find? I see at least another one. CCC is not palindromic because CCC backwards is GGG, right? It's not the same, right? But good try, Shamila. Anyone else? Anything else here that looks like a palindrome to you? Okay, here's another one. If we can write it out, right? So CTTTAAAG, CTTTAAAG. Okay, so these are all palindromic sequences. If you weren't able to identify any, do you understand better now that I explain it, or still not really sure? It's okay if you're not sure, but I need to know what is it that you don't understand so I can re-explain it.
I think I understand it better now, but until you rewrite the whole thing, it may be a bit difficult to get the palindromes for me personally.
No, I
I
agree
think
with you,
I'm gonna
right?
need to rewrite the whole thing if I
Yeah,
want to identify it.
that's one strategy. And another strategy is, we can actually create our own palindromes. Maybe after you create a few, then you would be able to see the trend, right? Okay, why don't I give you guys like, I don't know, like a minute or two. I want you to create like a 6 nucleotide long palindrome for me. Okay, so go ahead, try that. If you have it, do share. Anybody has a palindromic sequence they want to share with the class? No? Okay. Well, maybe if I let you know the secret of creating them, then it's much easier. So I want 6, right? I say 6 nucleotide. Okay, listen carefully if you are not able to create your own. Okay, so you can start with any 3 letters you want. If I say 8 nucleotides, then you can start with any 4. Okay, so 6 nucleotides. Start with any 3 that you want. I'm gonna go with A, T, C. Right. Then you would find the complementary nucleotide for C, which is G. Complementary to T is A. Complementary to A is T. And that's how you create it. This is palindromic sequence. Okay, you can find a complementary strand, TAG, CTA. You see, now it's the same. Okay. If that's confusing, you can try this method as well. Start with any one that you like, any three, and then for the bottom strand, you write the same 3 letters backwards. And then now you just fill it in with complementary strand. Okay, that also works. I don't know if that helps or not. It takes some time to practice, of course, but essentially that's what palindromic sequences are. Okay, last call. Any questions before I move on? Oh, good, Francisca. Thank you for confirming. It's always good to get a little bit of a feedback. Oh, by the way, you probably will receive an email soon regarding like course evaluation, right? So please do take the time to complete those. We take your comments very seriously. We want to know what you like and not like about the online learning experience, for example, what kind of improvements you would like to see. Right? So we gather your feedback, anonymous feedback, and we use them to improve our delivery. So please take the time to complete it when you get the invitation, the link for the survey. Okay, let's try another example here, and we'll just do it together. Right? So I give you a strand. This is similar questions that you will see on the quiz or test, final test. I'm gonna give you a sequence, and then I'm gonna give you 2 enzymes. Maybe we have EQR1 here and HindD3. And there is the recognition site here for EQR1 and for HindD3. So if the above DNA sequence is treated with these restricted enzymes, how many fragments will be produced? Yes, Oshuko, that is a correct. Oh, actually, that's not quite correct. Okay, so since there's a question, let me go back to here. So there is a sequence here, GTAGAT. You want to know if this is palindromic? Not quite, because if I write it backwards, then it's CAT-CTA, right? So if I read it backwards like this, it's not the same as forward, right? Okay. So if you want to create it with GTA, you can write GTA, no problem. And then you can write GTA backwards on the second strand. And then you fill in the rest using complementary. So G is going CAT like that. Right? And now it's palindromic. Okay, you see that? Does that answer your question? Okay, great. So back to here, yeah, no problem. Back to here, we have this DNA sample, okay? So you basically want to look for the recognition site of EcoR1, right? So do you see GAATTC? Okay, so we're gonna scan through it, make sure you're looking at the right one, 5' to 3', GAATTC. So that's GAATTC right there. There is a recognition site. We're gonna find another GAATTC, and I don't see any. Oh, I do, I lied. There is another one, GAATTC, right? All right, so how many times is this thing gonna be cut by EcoR1? Well, it's gonna cut it once here and then once here, right? So that's 2 cuts. But we are treating it with HindD3 as well. Okay, HindD3. And HindD3 recognize AAG, CCT. Let me choose a different color here. And we're looking for AAG, CTT. AAG, CTT is right here. I think that's the only one. I do not see another AAG, CCT anywhere else. Okay, so that means this thing is going to be cut once, twice, 3 times. If you cut a DNA 3 times, how many fragments are we going to have? Right. If you have a DNA, you cut it 3 times, chop, chop, chop. How many fragments are we going to have?
4.
4 is correct, right? 1, 2, 3, 4. So the correct answer is 4. Do we understand this? Like I said, it's a complicated lesson, so please ask questions. Why 4? Do you understand why there are 3 cuts, Francisca? Do you understand why we cut it 3 times? Yeah, so if you have a DNA and you cut it 3 times, then you have 4 fragments, right? Okay, take a piece of paper, you cut it 3 times, and then you will see you end up having 4 pieces, right? That's pretty much why. Okay. So we are gonna insert the DNA into a plasmid, right? And this is gonna require some explanation here. Okay. So we want to take the DNA here, and we're gonna treat it with restriction enzymes. Okay. So let me show you here. This is the circular DNA, and it has GAATTC here. Okay, so we're gonna cut it with equal R1, and it creates these sticky ends. Which means over here, if you add ligase afterwards, then they're just gonna stick back together, right? Okay. And in order for that to stick together, they had to be cut with the same restriction enzymes. Right? For example, if you cut a DNA fragment with equal R1, you're gonna have sticky end AATT, which will only attach to TTAA, right? If you take a DNA fragment that's cut by hint 3 and try to stick it together with equal R1 fragment, it won't work because the AATT is not complementary to the TCGA, right? So even if you add DNA ligase here, it's not gonna stick back together. So if you open up a plasmid with equal R1, right, and you take your DNA fragments that you cloned from PCR, and you also treat it with equal R1, then they have the same compatible sticky ends. Right? You can see here the AATT on one end will now be compatible with the TTAA here. And similarly, the TTAA on this end will be compatible to the AATT on the other side. So when you mix the cut plasmid with the cut DNA, it's possible for the DNA to go into the plasmid and it closes up. Okay. And you add a little bit of ligase, then you will now have a recombinant plasmid with a foreign gene. And that foreign gene in our example is the insulin gene, right? So you feed this into the bacteria, and the bacteria is going to make insulin for us. Okay. Any question about this? The key is you have to treat the plasmid and the DNA fragment with the same restriction enzymes so that you create compatible sticky ends. Okay, and you also have to use restriction enzymes that create sticky ends. If they cut straight down, that creates blunt ends. That's not going to stick together. You're not going to be able to insert the gene that way. Okay, so that's that. So here is a basic plasmid, and we're going to have have to think about what's going on with this plasmid. So a plasmid has a origin of replication. Okay, this is where the transcription will take place. Okay, start of transcription. Okay, and then it's gonna, copy everything in the plasmid. And these plasmid nowadays, you can just buy them. They are made in the lab. Okay, they're no longer harnessed from bacteria. It's easy to build a circular DNA using other forms of biotechnology that we're not going to learn. And these plasmid contains various insertion sites. Okay, so these here are insertion sites. Okay, these are locations where you can insert your gene into the plasmid. Okay, so let's say you have a insulin gene, and your insulin gene is like this, right? Okay, and then you have some flanking sequence here. If you treat the flanking sequence with restriction enzyme like EcoR1, then you will be able to insert it here. Okay. If you treat it with PST1, then you will be able to insert here. If you treat it with BAMH, then you can insert it here. So there are multiple locations. Okay. And one reason why you need to have multiple insertion points is that because your gene is really long, right? And when I treat my gene with EcoR1, the ideal condition is you create a sticky end in the end that you will be able to then insert into the plasmid. But what if the recognition site of EcoR1 also exists within your gene, right? What if there is a GTTA ATC in the middle, right? Then when you treat the EcoR1, not only are you going to cut the flanking sequence, you also will cut your gene, and then it will be useless, right? Okay, so if that's the case, maybe you don't want to insert using EcoR1. Maybe you want to insert using PST1 instead. Okay, so these are additional options for inserting your gene just in case one of these restriction enzymes cut your gene. Okay, so you only want to cut it at the end. So you don't want to cut it in the middle. Okay, so that's that. Now, secondly, you're going to feed this plasmid to the bacteria. But how do I know if the bacteria actually take up the plasmid? Okay, some of them might not take up the plasmid. Okay, it refuses to pick it up. There's no way to tell. You can't look at them under the microscope or anything like that, right? So we're gonna need to have a selection marker. We call it a selection marker. Right? So something that will allow us to distinguish which bacteria has taken up the plasmid. Okay? So that's what these are. Okay, so we use antibiotic resistant genes as selection markers. Okay, so TETR means tetracycline resistant. Okay, and this is like ampicillin resistant. Okay, so if the bacteria takes up the plasmid and you try to grow them in the presence of tetracycline, they will be okay because they have the tetracycline resistant gene. Right? Otherwise, they would die. So let me give you an example here. So this is a bacteria that has taken up the plasmid. Okay, and that includes a foreign gene of interest. And within this plasmid, there is the selection marker, ampicillin resistant. So if you grow the bacteria without the ampicillin here, then you have a lot of growth. Okay, lots of growth. That makes sense. Okay, because there's just lots of nutrients in the dish, and then whether they have the plasmid or not have the plasmid, it doesn't matter. They would just grow tremendously. Okay. And then if you grow it in the presence of ampicillin, okay, most of them will die. Any bacteria that did not take up the plasmid will die. Okay, because if it didn't take up the plasmid, they would not have the ampicillin-resistant gene, and they would all just die. Okay, and that's good because it's a selection marker, right? That means whatever is growing are the bacteria that I want. I want to harness these bacteria and then keep on growing them, so that I can turn them into, insulin factories, right? So if you take a bacteria that doesn't have the plasmid, right, and you grow it without ampicillin, it's fine. They will grow happily. But if you play it on a plate that has ampicillin, everything would die. Okay, because there's no ampicillin-resistant gene in them, right? So that's that. So let's see. A gene is inserted into the plasmid using E-core R1. The plasmid is transformed that means you feed it to a tube containing E. coli. You culture all the E. coli on a plate containing tetracycline. What result do you expect? Okay. Do you expect all of them to die, all of them to grow, or only a small portion would grow? Okay, what do you think? Okay, thank you for participating. The correct answer is only a small portion will grow. Okay, so you insert a gene using E. coli R1, it goes in right here, and then you feed this to a tube of E. coli. Not all of the E. coli will take up the plasmid. Okay, some will not, right? So then in the tube, you would have two populations of E. coli, those with the plasmid and those without. With plasmid, they'd have taken it up, and then without plasmid. Okay, but they're all mixed in the tube. So if you take the whole tube and then you plate it, you grow it on this culture that has tetracycline in it, all these will die. Okay, and then only those with the plasmid will be able to grow. So you get this situation. Okay, you're not going to get like lawn colony, as they are called, as in like it covers the whole lawn, right? Okay, only a few colonies, and those are the one you want. Those are the ones that has the plasmid. Okay, this looks like the exact same question, but it's not, because this time we are inserting using small instead of equal R1. So what do you think happens this time? Okay, thank you for participating. In this case, all of it will die. All of it will die. How come? Well, because we're inserting using the restriction enzyme small one, and small one cuts right through the tetracycline resistant gene, right? So you're going to pop in your gene right here, which disrupts the tetracycline resistant gene. So whether the E. coli takes up the plasmid or not take up the plasmid, they will die in the presence of tetracycline. Okay, so look here. This one takes up the plasmid. This one does not have the plasmid. I hope it's obvious that these will all die, right? Because they don't have the plasmid, they will die. But even if they have the plasmid, they will still die in this case because the tetracycline gene is being interrupted by whatever you're inserting, right? So even they take up the plasmid, they won't be able to gain resistance to tetracycline, and they will also die. Do you understand that? Right? So it's not just about taking up the plasmid. It's about what kind of resistance the plasmid gives you, right? That's why there are usually more than one resistant site, right? So here you can have tetracycline resistance or ampicillin resistant. So if you insert a gene using BAMH, we can select with ampicillin. If you insert a gene with PST1, then we can select using tetracycline, right? So it just gives you more versatility, right? As to where to insert the genes and how you're gonna select for it. Okay, so we're just gonna end with doing a question here. Okay, so here is the same same plasmid. Okay, and we have some questions that we are going to go through. Explain how you will amplify the gene you're trying to insert. We talked about that already, right? We can use PCR, right? By designing the primers and then amplifying it. According to the plasmid map, what do these things represent? I told you already. Those represent restriction enzyme sites. Okay, and it's important to know where these sites are. So when you insert your gene, you know exactly where it is going, right? If you treat the gene and the plasmid with equal R1, then it goes in right here. If you treat them with BAMH1, it goes in right here. Okay. What are MR and TET-R? Those are antibiotic resistant genes that gives the bacteria ability to grow in the presence of these antibiotics, right? And they serve as selection markers. Now, we haven't talked about this gel thing yet. We'll learn it next time. What I do want to go through with you are these two questions. Okay, so let's see. Let's see. Okay, I'm going to put the plasma right here. Hopefully you can see it. Okay, so if you insert the gene by treating your DNA, your gene of interest, the one that you've amplified with PCR, with BAMH1, and then you transform into E. coli, what's going to happen if you grow them with no antibiotics? Okay, no antibiotics. What do you think? Long growth, some growth, no growth? What's going to happen? Well, no bacteria, no antibiotic means just nutrients. So it will be long colony, lots of growth, no problem. What about growth culture with tetracycline? Because we're inserting with BAMH, it's going to go right here, right in the middle of the tetracycline resistant gene. So whether they take up the plasmid or they don't take up the plasmid, they will die. No growth. And what about you give them ampicillin instead? Okay, so the ampicillin resistant gene is uninterrupted, right? So there will be some growth, some colony. Okay? And those would represent the bacteria that has successfully taken up the plasmid. Remember, when you feed your plasma to the DNA, to the bacteria, when you feed it to a tube of bacteria, not all of them are going to take it up. Okay, only a small percentage will take it up. The question we're trying to ask is, how do we know which bacteria has taken up the plasmid? Like, they will look the same, right? Like, even if you look at them under the microscope, there is no physical appearance difference between them. Okay, so how do you know which one has taken it up? That's why we need to have the antibiotic selection marker. Okay, you grow them into ampicillin. Only those that has taken up the plasmid will be able to resist ampicillin and not die and form colonies. Okay. How about this? E core R1. If I treat it with E core R1 and then I feed it, transform means to feed it to a tube of E. coli. No antibiotic. What kind of growth do we expect, people? Lawn colony, no growth, or some colonies?
No growth.
But there are no antibiotics, though.
Oh.
No antibiotics, just nutrients. Okay, so anytime you have no
Anytime
antibiotics,
antibiotics, always lawn colony.
if there's no antibiotics, that's right, there's always lawn colony.
Okay.
Okay, because there's nothing stopping them. They just grow, grow, grow. Okay, we're inserting using E core R1. That means the gene goes right here. What do you expect to happen with tetracycline? Will it grow? No growth? What happens? Grow or no grow?
Some growth.
Some growth, that's right. Because we did not disrupt the tetracycline resistant gene, right? So any bacteria that has taken up the plasmid, they're going to be able to grow. Right? Many will die because they didn't take it out, right? But some will grow. What about in the presence of ampicillin? Anyone else knows what happens? Grow or not grow? No grow.
No growth.
No grow, why not?
Because of the presence of the ampicillin.
But they have the ampicillin resistant gene, right? This one is not being disrupted, right? Right? Because you didn't put a gene in the middle of the ampicillin resistant gene. So as a result, right, you will still have some colonies. Okay. All right. Now I know this is like a lot of information and you might not fully understand it the first time you hear it. So I encourage you to watch the recording of the current section, but you can also watch the previous recording as well. I think it's pretty much the same thing, but then, maybe I was explaining it a slightly different way, in the recording, and maybe you'll get it more. But more importantly, if you don't understand, you can ask me questions. Okay, don't go on YouTube and try to find information, because I am already simplifying this for you. Okay, if you go and watch it on YouTube, they probably have extra information that you don't need to know, and it's going to confuse you further. Okay, so please feel free to ask me. We will finish the rest on Wednesday. Okay, thank you for coming, and I'll see you Wednesday at 12:30. That's it for today. Bye for now.